Life has evolved from simple unicellular organisms to multicellular organisms, to plants, insects, pigs, and to the fascinating complex, and in our case beautiful, human beings. Essential to this evolution is cell adhesion. The cells of multicellular organisms not only have to structurally stick together, but also know when to stick together and when not to stick together during development. Cell adhesion is essential for cell polarity, motility, cell-cell communication, and enables a cell to respond to its environment. What parts of eukaryotic cells enable this to happen? Well, the eukaryotic genome encodes for proteins that are involved in cell adhesion processes, and they are also integrated into many signal transduction pathways. Cell adhesion also regulates the organisation of the cytoskeleton and adherence to the extracellular matrix. The proteins involved in this are called cell adherence molecules and are generally transmembrane proteins with intracellular and extracellular domains. Like any protein, they can be regulated allosterically. As you can see in this diagram, the orange ligand is inducing a conformational change to the yellow protein. This allows the yellow and green proteins to interact. Coming up next on the screen is an example of two transmembrane proteins interacting and inducing a signal transduction pathway. This is an example of how cell adhesion can enable a cell to respond to its environment. The pink ball in the cell on the left represents a second messenger and can be seen moving in the cell in response to the ligand outside of the cell. The importance of cell adherence mechanisms is demonstrated in mammalian fertilization. The male gamete, the sperm, has a specific structure which helps it to fertilize the ovum. Pertinent to cell adhesion is the acrosomal cap, which is an organelle on the anterior of the condensed sperm head. Localized here are the proteins represented by the G and S in the diagram. G is GAL-T and S is SED1, the function of which will be revealed soon. Represented here is the oocyte, the female gamete. Specific to cell adhesion here are the zona pellucida proteins ZP1, 2 and 3, and the purple balls, which are the cortical granules. The fertilization process begins as the sperm navigates its way through the corona radiata. The sperm then comes into contact with the zona pellucida. The SED1 proteins interact with the ZP proteins, docking the sperm and bringing it into close proximity with the zona pellucida. The GAL-T protein and ZP protein are now able to interact and initiate the acrosome reaction. The acrosome reaction is a signalling cascade which results in calcium-induced exocytosis of the acrosomal vesicles. These vesicles contain proteolytic enzymes such as acrosin, which are able to lyse a hole in the zona pellucida. At this point in time, as the vesicles are released, the inner membrane of the acrosome are exposed. This has receptors with an affinity for ZP2 proteins instead of ZP3 proteins. This enables a transient adherence between the sperm and the zona pellucida while part of the zona pellucida is being digested, and enables the sperm to still stay attached to the zona pellucida as it penetrates the zona pellucida and enters the perivitellin space. The sperm and oocyte membranes can now come into contact for the first time, and while their mechanisms of fusion are still being investigated, the protein izumu, which is a member of the immunoglobulin superfamily of cell adherence molecules, is located on the equatorial region of the sperm. In our diagram, it is represented by an eye and has been proven to be essential for fusion with the egg. On the oocyte, a tetraspanin called CD9, located on the microvilli of the oocyte plasma membrane, has been shown to also be required for sperm and oocyte fusion. The membranes can now fuse, and this induces a second messenger calcium signal transduction pathway, which initiates the release of cortical granules from the fertilized egg. This happens in a wave over the egg, and the granules move through the perivitellin space to the zona pellucida, where they then cleave the zona pellucida proteins, which changes the affinity of those proteins for other sperm. This ensures that they can no longer adhere to any other sperm, and so no other sperm can enter. This is termed block to polyspermy. Now a gamete is formed, and cell division leads to formation of a unique individual.